Neurological Disorders Lesson 2.3, How Fast Do Your Neurons Signal? The goals of this lesson are to introduce myelination and how it increases the speed of conducting the action potential down the axon, and to explore the consequences of disrupting myelination. At the end of this lesson, students should be able to explain why neurons need to signal quickly, describe how neurons increase the speed of the action potential traveling down the axon, and describe why myelination increases the speed of the action potential traveling down the axon. We'll achieve our goals with a ruler drop test, data collection and analysis, Socratic discussion, and an engineering session. To prepare for this lesson, you'll need to review the key scientific concepts that we were presented in it. Those concepts are that glial cells are the other cellular building block of our nervous system, and that myelination increases the speed of the action potential by addressing two constraints on conduction speed, and does this by preventing leakiness and saving energy. The consequences of damage to myelin, and the development of myelination. You can review this key scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of lesson structure, including instructions on how to manage the activity, and the student workbook provides additional explanation for your students. You also need to be sure to print the Do Now worksheets for your students. There are two versions of the worksheet, one in which students solve the formula D equals 1/2 AT squared, where they're solving for T, and the other that uses a table where students look up their reaction times based on the distance the ruler dropped. Use the version that you think will work for your students best. This lesson also requires centimeter rulers that are wooden or otherwise weighted. The key point of the lessons Do Now is to engage students with the idea of how fast our neurons signal. We'll get there by having students perform a ruler drop test for reaction time. Students will collect and analyze data. You'll give each student a copy of the Do Now worksheet and have them complete it with a partner. The worksheet instructs students how to complete the ruler drop test and then how to use the data they collected to calculate their reaction time and neuronal signaling speed. This activity takes most students around 15 minutes to complete. The key points of the discussion are that neurons signal very quickly, and to introduce the constraints of the action potential conduction speed. First, that membranes are leaky, which leads to the decay of signals, and second, that by adding more channels, we'd increase the energy requirements for our neurons. We'll get to these key points by analyzing data from the ruler drop test and with a Socratic discussion. The discussion starts with a review of the Do Now activity. Using slide 3, you'll review how students calculated their reaction time and compare their calculated reaction times with national data for 16 to 19-year-olds. You'll then use this data to calculate how fast our neurons signal. To do that, you'll first need to estimate how far their neuronal signal traveled, which you'll estimate on slide number 4. Then, on slide number 5, you'll complete the calculations to determine speed of signaling, which estimates to 9.4 meters per second. However, scientists have measured the speed of the action potential to be as slow as 0.5 meters per second. And that is quite a bit slower than our calculated 9.4 meters per second. So something's amiss here. Now the question becomes, what increases the speed of axonal signaling? And why does the action potential travel so slowly down our axons? Slide number 6 allows you to introduce the constraints behind why the action potential can only move so quickly down the axon. First, the membrane is leaky, so signals decay, and second, with more channels, which would prevent signal decay, axons would need to use a lot more energy to maintain their membrane potentials. The key points of the activity are to demonstrate that myelin addresses both constraints on the action potential conduction speed, because myelination prevents leakiness and saves energy. Additionally, we'll explore the consequences of damage to myelin. We'll get to these key points with an engineering session and Socratic discussion. In the activity, you'll have students work in small groups to brainstorm ways to speed up axonal signals. A word of caution. Students who have read ahead in their workbook or have had classes on the nervous system before might not be as stumped as students who are thinking about this problem for the first time, because they'll already know from past experience that myelin increases the speed of axonal signaling. For these students, you can change the question and ask them not what increases the speed, because they know that myelin does, but instead ask them how myelin increases the speed. After the students have had time to brainstorm ways to address both constraints, you'll lead a discussion about their solutions, which could be quite varied, and trust me, some of them might definitely challenge your own biological understanding of the system and its constraints. After discussing the student solutions, you'll use slides 8 through 10 to introduce the biological solution, myelin. First by showing that myelin solves both problems. It prevents leakiness and it saves energy. Then by showing how myelin wraps around our axons. And finally by showing how much myelin can increase the speed of neuronal signaling. This leads to one of my favorite questions to ask students. Which would you rather have, myelinated or unmyelinated axons? Students always answer myelinated, which in most cases is going to be superior, but this allows you to challenge them to think about circumstances where it might be beneficial for a signal to travel more slowly. And after being challenged in this way, most students do come up with the example of pain, and in fact some, not all, of our pain neurons are unmyelinated, which explains why sometimes we know something is going to hurt before we actually start feeling the pain. The last thing in the activity is to discuss what happens when myelin gets damaged, as it does in the disease multiple sclerosis. Multiple sclerosis will be discussed again in lesson 2.5, but since we're introducing myelin here, it's a good time to have students try to predict symptoms patients would experience if their myelin was damaged. In fact, MS patients experience a wide variety of symptoms depending on where they've lost their myelin. And again, we'll explore this further in lesson 2.5. The key point of the wrap-up is that our brains are not fully myelinated until the age of 20. We'll get to this key point by examining myelination data and a reflection and discussion. In the wrap-up, you'll present data that shows how myelin is gradually added as we age. After the students have analyzed this data, looking specifically for which areas of the brain are last to get myelinated, you'll present the argument that some scientists are making about this data. Because the data shows that the human brain, specifically the frontal lobe, is not heavily myelinated until the age of 20, some scientists argue that teenagers show poor judgment because their brains aren't fully myelinated. You'll then ask the students what they think. Do they agree? Do they disagree? Could there be another explanation? Do they have any additional evidence to back up their claims, one way or another? The key point of the homework is to use data on the development of myelination to make an argument. We'll get to this key point by writing a response paper addressing the argument some scientists are making about teenagers' poor judgment being caused by lack of myelination. This lesson is fairly straightforward, especially for students who have covered myelin in previous classes. That said, one of the most common questions teachers get is, why don't we all have the same reaction time? The answer here lies in the fact that reaction time improves with practice, especially on a test like this. Also, distraction increases reaction time, meaning if you're distracted, it takes you longer to respond, which is why so many states have now passed laws about not texting while driving. And finally, as we age, our reaction times tend to increase, meaning that older individuals take longer to respond than younger individuals. All that said, a really fun addition to this lesson is to have students repeat the reaction time test using subjects of different age, as well as in conditions in which subjects are distracted, perhaps by texting, to actually gather the data on how much distraction can impact our reaction time. At the end of this lesson, students should appreciate the importance of myelin in the functioning of our nervous system. Students should be able to describe how myelination increases the speed of the action potential conduction down the axon, as well as to predict the consequences of damaging myelin. An understanding of myelination explains the causes and symptoms of some of the disorders that we'll be explaining in lesson 2.5. Additionally, the development of myelination as we age is one of many examples that we'll see of how our brains change as we age. And don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members, and we'd be more than happy to help you.